The overall goal of this extracellular flux analysis is to assess the metabolic characteristics of naive M1 and M2 polarized bone marrow-derived macrophages.
This method can serve as a framework for investigating how particular cytokines, compounds, or genocodes affect the metabolic phenotype of macrophages. The main advantage of this technique is that it requires only a small amount of macrophages. Moreover, it measures all relevant glycolysis and mitochondrial parameters in one single assay.
Though this method provides insight into the metabolic phenotype of bone marrow-derived macrophages, it also can be applied to other types of macrophages or immune cells.
We had the idea to publish this method in JoVE since we frequently receive requests from other scientists to assist them with their metabolic extracellular flux assays.
On day 8 of culture, verify the presence of mature macrophages by visual inspection, and incubate the mature bone marrow-derived macrophages in 10 milliliters of citrate saline for 5 minutes at 37 degrees Celsius. When the cells have detached, wash the culture with 10 milliliters of PBS and count the number of viable cells. Then seed 5 × 10⁴ cells per well in an XFE 96 cell culture microplate in 100 microliters of culture medium per well, holding the pipette at an angle about halfway down the side of each well to dispense the cells. Add medium alone to the background correction wells A1, A12, H1, and H12. Then allow the cells to settle at room temperature in a cell culture hood for 1 hour. After confirming adherence, culture the cells in a 37 degrees Celsius 5% carbon.